Hey everyone, and welcome to AllAboutRx. I am absolutely thrilled to have you here for today's explainer. You know, as pharmacy students, you are going to spend a massive amount of time studying exactly how drugs interact with the human body. But here's the fundamental truth, the real hook for today's session. You simply cannot understand how a pharmacological drug works if you don't understand the very cell it acts upon, right? The cell is the ultimate target. It's the primary building block of the entire body. And today we are going to completely master its physiology.
Okay, so here is our academic blueprint for today. We're breaking this down into 4 main areas. First up, Section 1, the barrier. Then Section 2, the machinery. Section 3, the code and replication. And we'll wrap up with Section 4, the environment. Let's get right into it.
Section 1, the barrier. This is the cell membrane. It's this incredibly thin protective sheath, literally just 7.5 to 10 nanometers thick, and it's the very first hurdle any drug has successfully navigated.
Okay, let's dive into this. The accepted structure we use is the Fluid-Mosaic model, which Singer and Nicholson proposed way back in 1972. The core of this is a lipid bilayer. Think of it kind of like a sandwich. The phospholipids have a head portion that's hydrophilic, meaning it absolutely loves water and dissolves in it, and a tail portion that's hydrophobic, meaning it's fat-soluble. Because these hydrophobic tails meet right in the middle, the membrane is basically a wall against water-soluble substances, but it's totally open doors for fat-soluble ones. When we talk about drug delivery, this solubility factor is absolutely paramount.
Now, for your physiology exams, you will definitely want to commit these numbers to memory. The membrane composition is exactly 55% proteins, 40% lipids, and 5% carbohydrates. Those lipids include things like cholesterol, and its highly fat-soluble steroid nucleus basically dictates how permeable and fluid the membrane is, along with sphingolipids, and those sphingolipids are absolutely crucial in nerve cells for proper signal transmission.
Let's do a quick recall on that massive 55% protein chunk we just mentioned. You've got integral proteins, and these guys span the entire lipid bilayer. They go all the way through, acting as transport channels, carriers, and crucially, as receptors for water-soluble drugs, like peptide hormones, that can't just slide right through the fat. Peripheral proteins, on the other hand, attach to just one surface. They usually act as enzymes or help control transport.
Moving on to Section 2, the machinery. We're talking about cellular organelles. Once a drug actually passes that membrane, it enters the cytoplasm. And it's not just a random soup in there, it's a highly organized, incredibly efficient factory packed with specialized organelles.
Now, what's really interesting here are the mitochondria. We always hear them called the powerhouses of the cell, right? They extract energy from nutrients to synthesize ATP along those folded inner membranes called cristae. But from a pharmacological perspective, they do so much more than just make energy. They act as a vital storage unit for calcium ions, and they play a massive role in apoptosis, which is programmed cell death. They actually trigger this by releasing a protein called cytochrome C, which then activates the caspase enzymes that systematically dismantle a dying cell. It's a highly controlled demolition.
And this brilliantly illustrates the cell's post office, the Golgi apparatus. It's typically made up of 5 to 8 flattened sacs and has 2 very distinct sides. Materials arriving from the endoplasmic reticulum enter at the cis face. Once inside, these glycoproteins and lipids get modified, processed, and essentially slapped with a chemical shipping label, like a phosphate group, to ensure proper delivery. Finally, they exit through the transphase, neatly packaged as secretory vesicles, secretory granules, or even lysosomes. It really is the ultimate shipping department.
So where do all those raw materials come from in the first place? Well, that would be the endoplasmic reticulum, or ER. The ER is this vast, sprawling network of membranous cisternae. Now, when it's studded with ribosomes, we call it the roughed ER. And ribosomes themselves are these granular little structures made of 35% protein and 65% ribosomal RNA. Working together, they are the literal protein factories of the cell, continuously translating genetic codes into enzymatic, hormonal, and lysosomal proteins.
Here is your next key takeaway. And honestly, this is a massive concept for pharmacokinetics. While the rough ER handles protein synthesis, the smooth ER, which, as the name implies, completely lacks ribosomes, handles lipid synthesis. But most importantly for us as pharmacy students, it handles detoxification. The smooth ER is the cellular machinery directly responsible for detoxifying harmful substances and drugs. So later on, when you're studying drug metabolism in the liver, you really need to remember the smooth ER.
Integrating all of this incredible machinery is the cytoskeleton.
And, you shouldn't think of this as just a static, rigid skeleton. It's a highly dynamic, constantly shifting network of microtubules, intermediate filaments, and microfilaments that continuously assembles and disassembles itself. It dictates the cell shape, enables cellular movement, creates internal railway tracks for intracellular transport, and is absolutely essential when it's time for the cell to divide.
Of course, every efficient factory needs a cleanup crew. Let's compare lysosomes and peroxisomes. Lysosomes actually break off from the Golgi apparatus and come packed with up to 40 different hydrolase enzymes. Their job is to split organic compounds by adding water, which digests damaged structures and destroys unwanted bacteria. Peroxisomes look pretty similar, but they contain oxidases instead. They use hydrogen peroxide to oxidize and detoxify poisonous substances. Want a perfect clinical example? Peroxisomes in your liver cells are exactly what convert toxic alcohol into acetaldehyde. Let's move to section 3 and see how all this builds, the code and replication. We are now diving into the ultimate command center, the nucleus, to explore the cell's genetic blueprint and exactly how it goes about reproducing.
The nucleus is enclosed by a double membrane full of pores, and it regulates literally all cellular activities by turning specific genes on and off. Deep inside, suspended in the nucleoplasm, is the nucleolus. That's a dense structure where RNA is transcribed and ribosomes are actually assembled. But the most critical function of the nucleus is storing our entire genetic blueprint across 46 chromosomes, or 23 pairs in our somatic cells. When this genetic code suffers a mutation, whether single or multiple, it can lead to some devastating genetic disorders like sickle cell anemia or cystic fibrosis.
Now I want to slow down for just a second here because grasping gene expression is absolutely vital. It happens in two very distinct steps. First up is transcription. This happens inside the nucleus where the genetic information from DNA is copied into messenger RNA, or mRNA, and this relies on an enzyme called RNA polymerase. Second is translation. This is where the mRNA leaves the nucleus, travels out into the cytoplasm, and its sequence is decoded at the ribosome. It reads three-letter codes called codons to build a highly specific protein. So DNA to mRNA is transcription, mRNA to protein is translation. Got it?
So, the crucial point here is understanding how cells actually pass this code on through cell division. We have two distinct types. Mitosis divides a parent cell into two identical diploid daughter cells, each maintaining those 46 chromosomes. This process is all about growth, tissue repair, and everyday maintenance. Meiosis, on the other hand, results in 4 haploid daughter cells, and each of these has only 23 single chromosomes. This strictly occurs in reproductive cells, the gametes, and is purely for sexual reproduction.
Mitosis itself unfolds in 5 rapid-fire stages. Let's run through them. Prophase, the nuclear envelope breaks down. Metaphase, the chromosomes align right at the center. Anaphase, the chromatids separate and get pulled to opposite sides. Telophase, the nuclear envelope begins to reform. And finally, cytokinesis, the actual physical division into 2 distinct cells. It's really important to note that when these highly regulated mechanisms fail, you get rapid, abnormal cell division, which we clinically recognize as cancer. Finally, we arrive at section 4, the environment. We're zooming out a bit to look at cellular fluids. Remember, a cell doesn't just exist in a vacuum. It is constantly interacting with the fluids surrounding it. And understanding these compartments is absolutely essential for understanding how a drug distributes throughout the whole body. We primarily deal with 2 compartments: extracellular fluid, which includes the blood plasma and interstitial fluid outside the cell, and intracellular fluid, also known as cytosol, which is inside the cell. Consider the drastic difference in major ions here. The outside fluid is absolutely loaded with sodium and chloride, but the inside fluid, it is packed with potassium and phosphate ions. The body actually spends a lot of energy using special mechanisms to maintain this exact concentration gradient. Also, think about the volume. Intracellular fluid makes up a massive 2/3 of total body water. It provides the literal medium for all of these complex chemical reactions we've been discussing today. Okay, so we've seen how perfectly, meticulously structured the cell is, right? From its highly selective lipid barrier to its protein manufacturing ribosomes, all the way down to its tightly regulated genetic code. Which brings me to a somewhat provocative question I want to leave you with. If the pharmacological drugs you will soon be dispensing are specifically designed to target these highly specific cellular structures, what happens when a systemic disease, or say a virus, actively hijacks the cell's own machinery to work against it?
I really want you to think about that as you review these fundamentals. Thank you so much for joining me for this explainer here on All About Rx. Keep up the great studying, and I'll see you in our next session, where we tackle exactly what happens when pathology disrupts this incredible physiology.